Okay, now let's do a couple of neural circuits. So let's say that I want you to do a neural circuit that causes a sea anemone, a polyp form of a cnidarian, to retract its tentacles if it's pinched, if it's damaged, or if something pinches against it. Okay, so let's draw a polyp, and as a general rule, to answer these kinds of neural circuit questions, you want to think about what's the starting condition, starting input or condition. That's a pinch. And then somewhere else on the page, maybe down on the right, do the ending condition, retract. That's the behavior you want. You can start at either end with the pinch. So you might start at the pinch and realize, that would be a sodium stretch channel, would be a good way to do that. And at the end, retract, that would be longitudinal muscle. Remember, longitudinal muscle is running lengthwise, both down the tentacles and all along the sides. So longitudinal muscle. And now you need to connect those two together. And you can start at either end, and I'll show you by starting at this end, the end of it instead of the beginning. So we're going to need a neuron, a motor neuron, that can cause all of these to contract. We can do it all with one neuron. I could connect this one neuron over to the other side, or if I wanted to, I could do it with two neurons. They'll both work, and for neural circuits, you just need something that'll work. And let's put letters on these. Okay. So now we have a way, if we cause action potentials in A and B, that will send action potentials to these muscle cells. Those muscle cells will shorten. Each muscle cell will shorten. And if all of them get shorter, then our whole anemone is going to shorten, it's going to contract. Okay, so how do we do the pinch? Well, we're going to need a neuron that detects a pinch. So here's something doing the pinching. And we want stretch channels in there. Okay, so we'll have sodium stretch channels. And we want that to be on a part of a neuron. And here I'm going to draw a loop. You don't really have to do that. And we'll call that neuron C. Okay, now we need a way to connect those two together. And in this case, all we want is for our pinch to cause contraction. So all we need to do is have a connection that goes directly to these two. And now let's check. Will it work? We pinch, we open those stretch channels, sodium comes in, that sodium gets to the cell body or starts an action potential out there and a lot of sensory neurons. The action potential travels down. It generates a new action potential in both A and B. The action potentials get out to our longitudinal muscle and they contract. Our circuit is going to work. Okay, let's make it more complicated. Let's imagine that we want that circuit to contract only if there's a pinch but no food. But if there's a pinch but at the same time there's some little organism nearby that's leaking amino acids, potential food, then we don't want to retract. We want to stay out there as an anemone so that we might catch it. So now we have a new circuit. We want pinch, retract, pinch, plus some chemical, let's say amino acids leaking from that little potential food item, pinch plus action potential, don't retract. And now down at the end we have retract or don't. Again, we draw our anemone. Let's put a few longitudinal muscle cells in it, enough to represent what we're doing. We know we're going to want retraction, so, and this time I'll draw it with just one cell, just to show you it can be different. We have our sensory neuron here. This time I'll call it B. Here's our sodium stretch channel. We have a pinch, and that will cause contraction. That'll cause A to stimulate these longitudinal muscles and cause contraction, but we want an inhibitory as well. If we have a little creature here that's doing the pinching, this is a claw, okay, but our little creature can't help but leak a few amino acids. All aquatic animals are always leaking a few valuable molecules or other odorant molecules. And so let's have another cell here. Another cell, this time C. And on this one, we're going to put a chemoreceptor protein that binds amino acids, or at least one amino acid. And when it does, that activates some pathway that allows sodium in. And so this neuron, neuron C, is going to be producing action potentials if there's amino acids. And let's make it inhibitory. Okay, so one pathway stimulates it, but if there's also amino acids, we inhibit it. Okay. What if we wanted it more complicated yet? What if I said I wanted it in the presence of a pinch and amino acids? I wanted to wave that tentacle around. So how would we, or at least move that tentacle? So let's just go with a simple movement of that tentacle. Here we are. Here's our longitudinal muscle. But now we want the potential to retract, but also to move the tentacle. So if we kept it simple, if we just contracted longitudinal muscle on one side of that tentacle, and if those shortened, we'd bend that tentacle. Okay, so again, here's A. Notice I'm only doing one axon from each cell. That's typical, one neuron, one axon. Okay, here's our pinch, stretch channel. This was B. Here's our stretch channel. That's stimulatory. And a receptor, C. That's inhibitory, but we also now want to move that around, so let's have another branch to a neuron D, and that neuron we want to cause the movement. So let's run through our circuit. Will it work? We have a pinch, open a stretch channel, that stimulates A. So if we only have a pinch, all that happens is we retract. We do an escape behavior. If we have a pinch plus amino acids coming from our beastie with the claw, then we inhibit the retraction and we stimulate bending. So these are ways to use neural circuits to cause all kinds of movements, and one of the things you should be doing is practicing how to do neural circuits with the different phyla, with worms, with clams, moving mouths, moving feet, moving whatever you want, with various inputs from sound, touch, chemicals, light, and so on.